So our next speaker is Ian Cheeseman from Texas Biomedical Research Institute, and he's going to be telling us about single-cell genomics. But it's not him.
Sorry, that's Simon then.
Hello. So I'm not Ian Cheeseman. I'm actually his postdoc, Simon Trevino. I'd like to thank everyone for this opportunity to share our work. Today we'll be talking about some of the progress that we've made on capturing single-cell genomes from patient infections. And right now we're analyzing quite a large data set, but I'll be able to show one infection in detail. Okay, so I'll just start with some really quick background. We've known for some time that patients can be infected with multiple parasites that have different lineages. Understanding this genetic complexity is important for having a better understanding of transmission, virulence, and the spread of drug resistance. Currently, it's not known just how many haplotypes are present in many of these patients or how they're related to one another. Single-cell genomics would give us a very good shot at understanding this parasite ecology within the patient, but due to technical difficulty, this has not been very feasible on a larger scale. So what are the kinds of information that we can get from single-cell analysis? Everyone here, I'm sure, is fairly familiar with bulk DNA studies, and this is very helpful. We can pick up on interesting mutations, and if we sequence deeply enough, we can even see more rare mutations. But we can't take bulk DNA and derive out the individual haplotypes present in that patient. So we lose the genetic context of that mutation, and we're still left not knowing what the actual true diversity is within our samples. In addition to haplotypic diversity, single-cell analysis can tell us a thing or two about recombination. So in this cartoon here, I've drawn a very simplified scene that might happen in a mosquito. So here we have two mosquitoes. I've colored shared variation between haplotypes, the same color. And so in mosquito 1, we have clonal haplotypes. In mosquito 2, we have haplotypes that have different lineages. And in the mosquito, they'll undergo meiosis. If both of these mosquitoes infect the same host, they'll deliver their offspring. And if we look at the shared variation, which is called identity by descent, we'd find that for haplotypes that are derived from mosquito 1, if we compared them across the genome, we'd have 100% IBD. In contrast, if in mosquito 2 we had two separate lineages, we would only have 50% of IBD present, represented in the blue and orange in the center here. And I could go ahead and draw more rounds of meiosis, and we would see that 50% number drop. And of course, if we compared a haplotype from mosquito 2 to mosquito 1, we would see 0% IBD. And so this is a very kind of clever way of learning the recent ancestry of parasites. It's not so applicable to long-term ancestry, however. With these kinds of questions in our mind, we decided to develop a single-cell genomics workflow based on facts. And in this method, we have our patient samples. We purify out the white blood cells, ship them on ice, and thaw them in the lab. And so once they thaw, only the ring stage parasites survive. We grow them for a short time, not allowing for reinvasion of new parasites. Then we can dye the DNA with a fluorescent label and sort them by a standard flow sorter, one cell to a tube. And since the amount of DNA we get per each cell is very small, a ring might have about 24 femtograms of DNA, we absolutely have to rely on some kind of amplification step. So in this case, we use multiple displacement amplification and have seen good results with that. Once we have enough DNA, we can go ahead and prepare our library in a very standard fashion and do our whole genome sequences. And I should point out that this, we go through some lengths to ensure that we can resist contamination as much as possible. A lot of the preparation of reagents and molecular biology is actually done in 2 separate hoods. And in fact, one of these hoods is behind an additional plastic barrier. We also use dedicated PCR machines for different steps so that we don't have any chance of cross-contamination. And recently, we found a few more modifications improved our genomic data quality. For one, we've chosen to begin growing for 40 hours instead of a shorter amount of time. And this allows for the single cells to clonally replicate within the same red blood cell. Starting with a higher amount of DNA improves our MDA results. We also found that using sterile PBS for the capture buffer is important, as well as using QIAGEN's latest kit, which is designed just for single cells without library amplification. And all of those details can be found on bioRxiv that was put online this summer. To give a real quick sense of the kind of improvements that we've made, this is a typical FACS plot of an infection that's asynchronous. So we have all the life stages represented here. If you were to grow your sample for a shorter amount of time and sort only the rings and perhaps early trophs, you'd only get about 50% genome coverage. However, if you let them grow longer, as we're doing now to 40 hours, and gate only for the Schizonts, you get about 90% genome coverage. This is really excellent for downstream analysis, and it compares actually to taking bulk DNA, which we typically get about 95% of the genome at a usable depth of 5, read depth of 5. So with an improved method in hand, we decided to analyze a challenging set of infections from Chikwawa, Malawi. And in Chikwawa, there is a high transmission rate of falciparum. The average person is infected every 2 days. Analysis of bulk data showed us that we would expect to see both monoclonal and polyclonal infections in this area, so that's super interesting from a biology standpoint. And here's a summary of what we have so far, and this should give you a sense of the robustness of our method. In the gray are single cells that made it through the entire workflow and ended up in the final analysis. In green are cells that just did not yield as much genome coverage as is usable, and in blue are mixed genotype calls, which are tubes that we suspect the FACS misfired into, giving 2 or more cells. And I will point out that this outlier was actually used with a previous method, so we have more cells with less coverage that weren't usable. So if we kind of omit that, we can see that we're getting very usable data, 8 or 9 out of 10 tubes independent of infection. In the remaining time, I'll just go over one complex infection in some detail. So on the left is a classic pairwise UPGMA tree. All these dots are individual cells that we sequenced. There's about 43. And the first thing you see is that there's about 7 haplotypes within this one patient, and they're present at different frequencies. So our yellow haplotypes are about 40% of the infection, but we have as few as just 1 out of the 43. So a very low representation of that haplotype. With the number 7 haplotypes in hand, we can borrow an analysis from ecological science, a rarefaction analysis, and figure out whether or not we've sampled our infection deeply enough. We just had happened to try 43 cells as a start, but we find that that's actually enough to cover most of the diversity in this infection, so we can stop there. I should say that for the other 15 infections, we're finding that about 24 single cells is enough to cover the diversity. However, we have seen some where we're going to sequence more than 50. The nice thing about this method is that you have these single cells in your freezer, so if you have to go back and analyze more samples, you can just go to the freezer and run some more of the QIAGEN kit. And so we're also interested in IBD, and just to remind you from the beginning of the talk, here's a cross where if you were to trace the amount of shared variation, you would see a block of 50%. We can do this with our data as well. So in the center here are the 7 haplotypes that we got from this one patient. And this trace here is just IBD analysis of 2 of these chromosomes. And so we'll see on the y-axis are differences in SNPs. And we'll see that there are these large blocks of shared variation as well as large blocks that are not. And so we would guess that this underwent a very recent meiotic, these 2 parasites underwent kind of a sibling meiotic recombination. And so if instead of just looking at chromosomes, we look at the entire genome and do this for all samples compared to one another. On the y-axis here, we have total length of IBD. On the x-axis, we have the mean length of those blocks. Then in the aggregate data, we begin to see classes of parasites emerge. And this is kind of in agreement with the pairwise tree that I showed you before. So we'll see clonal parasites that share variation across their entire genome, but we also see some that share quite a large portion, which we're calling siblings, as well as parasites that are unrelated. So we take this to mean that actually this patient was infected by 2 separate mosquitoes. However, there are caveats to that. And so this is great, but we want to be sure that we are actually analyzing something that's representative of what's in the patient. So we always run a control where we compare allele frequencies, in this case at 9,700 sites, from the bulk DNA that we get at thaw time point 0 to pooled single cell data. Our R squared is high, and so we're pretty confident that we're capturing what's there. So to summarize, we're now achieving 90% single cell genome coverage fairly routinely. We did this by targeting late stage parasites using sterile reagents and the QIAGEN FX single cell kit. In one patient, we see 7 unique haplotypes and have detected recombination sites across chromosomes. In this patient, we see both related and unrelated haplotypes. And we're currently analyzing over 400 cells, and more will come shortly. So I would like to thank people at Texas Biomedical Research Institute, especially in the Anderson Lab, as well as our patients and doctors, and we're funded by NIH and Wellcome Trust. And for further questions, I'll be at a poster session in a few hours, but I'll be there for I'm only available for a short time if you have additional questions. And with that, I'll take any questions you might have.
You said you had about 5x coverage throughout. What about
Yes.
the mitochondrial DNA and the apical?
We don't see mitochondrial, so not so much.
Have you tested directly ability to catch rare haplotypes, like if it's less than 1% of the total population, if you just do like 50 or so, it seems like you might miss those 1 out of 100 or 1 out of 1,000 haplotypes?
Yeah, absolutely. I think that's a tough experiment to do as a control, just because of cost. So if we made an artificial mixture and did 1 versus 1,000, we'd have to maybe cover like 100 cells. But as far as we can tell in the allele frequency data, it looks like we're capturing most of it. But yeah, those will, it becomes an issue of sampling, right? And we just have to stop at some point. But yeah.
Super. Cheers. Thank you very much, sir.
Thank you.